Hello again. Okay, to conclude this block, we're going to have 3 presentations on pragmatic and adaptive trials in organ donation and transplantation. What are pragmatic and adaptive trials? I'm not entirely sure. So hopefully someone on the panel will explain that to us non-scientists. So we're going to hear from Marat Slessarev, Dan Harvey, and Maureen Meade. I'm just going to introduce them all now so that we can move through things smoothly. Dr. Marat Slessarev is a clinician scientist and an adult intensivist based at Western University in London, Ontario. He joined Western faculty in 2017 as an assistant professor in the Department of Medicine and does his clinical work at the London Health Sciences Centre. His research focuses on sedation, neurophysiology, and cognition in critical illness. And Dr. Slessarev is the co-lead on the CDTRP endorsed project Neurologic Physiology After Removal of Therapy, or NEWPART. He also is one of my long-suffering supervisors, which is some of his most important work, I think. Dr. Dan Harvey is a consultant in intensive care medicine at Nottingham University Hospital and an associate professor at the University of Nottingham. He's chair of the UK Intensive Care Society's Legal and Ethical Advisory Group and an author of the GPICS guidelines. Dan has an active research interest with roles including National Lead for Innovation and Research in Organ Donation for NHS Blood and Transplant, Vice Chair of the NIHR's National Critical Care Group, and is Joint Chief Investigator for the SIGNET study, the world's largest interventional study in organ donation, which I assume is a pragmatic trial. Dr. Maureen Meade is critical care consultant at Hamilton Health Sciences Centre and a professor at McMaster University. Dr. Meade has spearheaded numerous national and international randomized trials. She's co-director of the Canada Donate Research Program and vice chair of the CIHR-funded Platform Trials Initiative of the Canadian Critical Care Trials Group. So thanks to everyone for being here. And Dr. Slessarev, we'll start with you.
Thanks, Nick, for that great introduction and the spirit of mentorship. Sorry? Oh, yeah. Sorry. I forgot about that. Forgot I'm wearing a mask. Thanks for that introduction. And it gives me great pleasure to be starting off here. In the spirit of mentorship, you know, I've been mentoring you, but Dr. Maureen Meade is my mentor. And so it gives me a great pleasure to speak here. I've only been given 5 minutes. It seems pretty unfair. So in those 5 minutes, I'm going to hopefully tell you 3 reasons for why I think we need to do pragmatic and adaptive trials in organ donation and transplantation, and then why I think CDTRP is well positioned to lead this work in Canada. So I have some disclosures. I have funding for research from the following organizations as well. I receive a stipend for my work as a regional medical lead for donation at TGLN. All right, so the first reason is that observational studies are important in informing what we do in clinical practice, but they can be misleading. And case in point is the use of vitamin C in sepsis. And this is a very important concept that came up in critical care worlds. 5 years ago, there was a study published before and after design at one center with 47 patients enrolled that told us that only if you give vitamin C to your patients with sepsis, you can drastically reduce mortality. And with the help of social media and podcasts, this became clinical standard almost overnight across centers in North America and the world. Fast forward to 5 years from that point in time, we have numerous trials that didn't show benefit from vitamin C. And most recently, a trial was published in New England Journal that was led by the Canadian researchers that enrolled patients from 35 centers internationally, 872 patients randomized to intervention and control, which definitively showed harm in the intervention arm. And so something that was standard practice based on observational work was actually shown to be harmful to patients and hopefully will no longer be a standard of care in our units. Now, there is paucity. The second reason is there is paucity of good quality evidence regarding donor management and donor interventions. And for NDD donors, the Management of Neurologically Diseased Organ Guidelines issued 36 recommendations and good practice statements. Only 2 out of 36 had moderate quality of evidence, and there was few RCTs to inform practice. In donors, in DCD donors, the question of ante-mortem administration of heparin is a huge controversy because we have 55 observational studies, but not a single RCT. Those observational studies, like the vitamin C story, tell us, hey, it's a great thing to give heparin to donors to improve outcomes in recipients. But when we subject that data to methods such as meta-regression, that benefit disappears. And so the effect is unclear. And so we need RCTs to inform our practice. The third reason is RCTs in this field are feasible, but they require appropriate frameworks. In 2015, a randomized controlled trial was published that demonstrated that therapeutic hypothermia cooling NDD donors can reduce delayed graft function in kidney recipients, so should be really implemented as a good intervention. However, the trial did not consider organ recipients research participants and did not seek informed consent. And so Public Citizen, which is a public advocacy group in the United States, picked on this. And this was a huge scandal that did a lot of damage to the trust in organ donation research. So as Dr. Murphy recently argued in his paper, in this case, science got ahead of the ethics. And so their call was that we need Canadian ethical guidance on how to do these kind of trials in this space. So those are the 3 main reasons why I think we need trials in this space. And I think CDTRP is well positioned to lead this work in Canada. We have diverse expertise across disciplines from theme 1 all the way to theme 5. And I love the exercise, right? It really woken me up for the afternoon session here. We have established partnerships that formed over years with our patients and donor families. And we have national representation and international collaborations with folks like Dan Harvey, who will speak after me today. And I want to conclude with saying, look, who benefits from RCTs? Well, obviously recipients, because they get the best graft. Donors, because they, by maximizing their opportunity to donate, we're honoring their wish and the wish of their families. The healthcare providers who are committed to making donation experience a success. And obviously the healthcare system, because we're not only going to be finding out interventions that work, we will also find interventions that don't work and should be de-adopted, just like the vitamin C example that I mentioned at the very beginning. So I'll end there and put a plug in for a dedicated 3-hour workshop starting at 7 a.m. tomorrow, running virtually, where we're going to be discussing this in more detail. Everybody's welcome to join. You can register on CDTRP page, and I'll stop there.
Yes, he did say 7 a.m. So if you don't mind, we'll just hold questions for the end so we can have a nice long discussion period. And so I'd like to welcome Dan Harvey for his talk.
Thanks very much. Can you guys hear me okay?
Yes, we can.
Excellent. Well, it's really a pleasure to be here virtually. I'm really disappointed that I can't be with you in Canada. I have had a fantastic experience with CDTRP in the past. In fact, it's not an exaggeration to say that the SIGNET study and much of the work that we've done in the UK was initially inspired by the work that CDTRP had done, not just scientifically, but also in the way you've brought donors, families, recipients and scientists together. I think that's something we've been looking to replicate in the UK And in one of those ways of doing that is with pragmatic studies, of which the SIGNET study is one. So I'm going to spend just a few minutes just to explain how we got that off the ground, and maybe where some learning points for us all thinking about collaborative, pragmatic, and indeed platform and adaptive studies in organ donation might go in the future. Really fantastic to share the stage with Marat and Maureen, both of whom I've just got the most tremendous amount of time for, and impressed by the work they've done already. And many people in the audience have been guiders and mentors of mine, and indeed serve in a formal role on the SIGNET study. So my great thanks to them, including Matt. Okay, so a little bit of disclosures from me. I get some money from NHS Blood and Transplant in my salary, but nothing else of relevance. You've heard the rest. So a little bit of outline what we're going to talk today. I'm just going to talk a little bit about what the facilitators and the barriers are for running national pragmatic RCTs in organ donation. Tomorrow, we're going to look at the detailed mechanics of the study design, capture, and so on in the operation in the workshop, and it just encourage everyone to come to that if they can. It's 7 a.m. in Canada. It's it's 3 p.m. in the UK, so it's my fault. It's so early. My apologies. I've just gone on. Yeah, and then in the future, I hope to come back and tell you the results of the study. We'll talk about dissemination and implementation. We heard a bit about that this morning and how vital that is. There's no point doing the science unless we can implement it right. But again, we're in a perfect position when it comes to pitching to funders like CRHR or NIHR in the UK about how not only we can do the science, but we can implement it almost immediately as well. I hope to be able to share some grant applications with everyone in the audience in the future and build those collaborative, pragmatic and platform approaches. So what is SIGNET? For those of you who haven't heard about it, statins for improving organ outcome in transplantation. We're taking 2,600 donors after the diagnosis of death by neurological criteria in major donating hospitals in the UK, and randomizing into a single enteral intervention of 80 milligrams of simvastatin versus protocolized standard care, which is blinded to the recipient teams. And the primary outcome is a composite of death, mechanical cardiac support, or renal replacement therapy in the first 30 days in cardiac recipients. Although importantly, we're tracking outcomes through all the other secondary outcomes in many other organ recipients. Something that of note was not really adequately done in that renal study, and is one of the reasons why we think that pragmatic and platform approaches are so crucial, because they track all of the outcomes of relevance in recipients, although they don't yet do so in donors and families. And that's something we'd very much like to talk about tomorrow in the workshop. So how do we get here? What are the facilitators and indeed the barriers for doing a big study like that in the UK? And it is, we think, by the time it is finished, going to be the biggest global interventional study ever done in organ donation. And so not a straightforward undertaking. Well, we had the right infrastructure. So we were lucky in the UK in having organ donation and matching retrieval all under one clinical operational roof in NHS transplant. And although blood and transplant, even though transplant is actually nominally devolved from that, it is very closely linked, most importantly through the UK transplant register. And we have an electronic patient record, for example, that tracks all donors and links to the transplant register, making data capture feasible for this sort of study. We had some really great cross-speciality collaboration in building it, not just myself and my co-investigator, Professor John Dark, who is a heart transplant surgeon from Newcastle, but many other co-applicants from other transplant donation organ fields. Great research networks we'll talk about in a little bit of luck with a funding call, and you do need some luck for this to work out. And you know, COVID was a disaster for UK ICU and for UK research, but it did help us organize the training and the setup for the SIGNET study. So, you know, there are some small benefits there. What were the barriers? Well, I think the regulatory framework for deceased organ donation, we've just heard a little bit about in Canada and the US. It's a little bit clearer in the UK, but there are still some grey zones, and we needed to navigate some of those, and we'll have some more to do in DCD. I'm absolutely sure. COVID was a big problem for us, obviously. Hospital research and innovation teams are sometimes not so interested in research and not particularly interested in innovation. So that is a challenge. Data management and GDPR in the EU setting, or how we can move people's data around, is a pressing challenge. So we just talk a little bit about those. What about the infrastructure then that helps us in the UK? And some of this is replicated in Canada. Bits of others we will need to line up, and that again is what the workshop is about. We had that clinical and operational system that I mentioned, which includes having consultant intensivists on every ICU, as well as specialist nurses who attend every potential donor. A clinical trials unit, which was built into NHS Blood and Transplant, as well as a research team, and that electronic national hub. So all of those things really just need tilting towards a pragmatic randomized controlled trial. We had some funded research already, an existing biobank in some of those level hospitals, for example, funded by NHS Blood and Transplant and the MRC, Medical Research Council in the UK. There are actually some other research units, but we had less to do with those, and there's an opportunity for the future, I think. We had, of course, that UK Transplant Register, which tracks the outcome of every organ recipient. And that's fantastic, although it is somewhat limited. So unlike the other studies that Maureen and others colleagues in the Canadian Critical Trials Forum have run very successfully, we're not able to actually choose the outcome we want, but rather pick from what's already available if we want to keep the costs to look to somewhere where an NIHR would fund us. So we can use that. It's essentially cost neutral. And there are some excellent resources as well inside UK critical care research infrastructure and network then as well. And I will just highlight the recent experience with pandemic studies, particularly REMAP-CAP, a study that was now run internationally, has over 350 centers and had randomized 10,000 patients. But was first opened in some UK centres, including my own, and has really taught us a great deal about what can be achieved with pragmatic and indeed platform approaches. We can randomize, instead of randomizing occasional patients with a condition, we can randomize the vast majority and get to an answer really quickly, and that will be something I'm sure Maureen will talk about more. I'll run short of time. So yes, cross-speciality collaboration. Here's myself and John at the 70s do at the British Transplant Society meeting. That's what collaboration should all be about. But it's been beaten into a hat there by the dancing I've just done in the kitchen, which has surprised the cat. So that's one thing that this conference will now be famous for, I think. You definitely need organ donation, critical care and transplant expertise in a group if you're going to build these sorts of pragmatic trials. They need some experience in research, clinical, and critically in governance, I think, and in the appropriate regulation. And they'll have the contacts right across the ICUs and the transplant centers, and also in the ODOs and the equivalents to try and make and tie all these different bits of data and individuals together, because it's a massive group that needs to be brought together to make this work. They can solve the problems that come up. The dancing is optional at the British Transplant Society, but clearly not at the CDTRP, and that's probably the way it should be. Of course, it wasn't just myself and John. Actually, we had this in the embedded CTU, the clinical trials unit, been fantastic. The stats expertise is completely crucial, but also how you physically move data around and transfer it between different organizations and in different groups, particularly in a blinded fashion. And we've integrated some of that with our electronic patient record and the UK Transplant Registry. There is more to do to automate that into the process in a platform study, particularly thinking about the quality of that data and where the risk in some of its quality may be, because it wasn't always designed for the reasons that we're using it. And clearly the study management has been crucial as well. The research networks, I'm going to highlight CDTRP as really showing us the way on how this could be done. We are some way behind, but we have a new one in the UK, ODTRN, which is trying to do similar things, and the UK Critical Care Research Group is also flatteringly modelled on the Canadian Critical Care Trials Forum. So NIHR in the UK are very strong, and there's a network in hospitals that allows us, and of course NHS blood and transplants clinical leads. But really, it's a combination of all of these things that you need to run pragmatic studies in organ donation. Bit luck with the funding call. Like I said, it was an ODT specific call to ask us to apply, although the funds weren't reserved for us. It was open competition with other specialities after peer review. But maybe a politics with a small p. Clearly, we were going through the deemed consent legislation process at that time, and it may be that the donation and transplantation were higher at the agenda than they would otherwise have been. I'm going to whip through this because we'll talk a bit more about this tomorrow at the workshop. But just to say, you need to be flexible. We need to be ready for the bumps in the road. So COVID waves were definitely bumps in the road for the SIGNET study, initially in getting some of our sites open, and then for the effect that they obviously have had on donation and transplantation in the UK and worldwide. So I think being resourceful and being nimble, and having, probably most importantly, studies that are designed to be pragmatically apply and attach to your existing ICU and organ donation processes, rather than mandate something completely novel, is the way to go, at least while we're starting out. Just going to talk about one barrier. We'll talk about those tomorrow, and that's regulation. So the MRHA Innovation Office in the UK, they're the body charged with looking after research safety in the UK, were once we got through to the right individuals, really fantastically helpful and really interested in what we were doing, and agreed, for example, that we didn't need to classify SIGNET as an interventional medicinal product for this purpose, which does change the regulations significantly. That our specialist nurses were already experts in consent and didn't need to be signed onto the delegation log, for example. That makes it practically much easier to roll out across more than 100 hospitals we're opening now. And that they're really the experts in taking consent from potential donor families, and an adjusted approach to safety, for example, recognizing that existing definitions of serious adverse events don't really apply in adequately in a donor population. So more talk about all of that tomorrow. Just to acknowledge those that have helped, not least many of you in the room, and of course the extremely generous donation from donors and families who are involved in the SIGNET study. To the latest date, we've randomized more than 400, and the study's going really well. So thanks very much for the invitation again.
Thanks very much, Dan, for sharing your experience, and congratulations on getting such an amazing study underway. However, you did go over time. Therefore, during question period, you'll be asked to do the Macarena in front of everyone on full screen.
It's only fair. And I'll take my...
Yeah, thanks. Okay, now I'm pleased to introduce Dr. Maureen Meade.
Sorry, I was muted. Okay, thanks very much, Nick and Marat and Dan, for your very kind words and introduction. And I'm just so pleased that we're holding a session such as this today. And I'll get right into it. Oh my goodness. Perhaps nobody but Fred will recognize this slide that he and I presented in 2015 to the CDTRP when we first joined. And we were talking about our research program, and our vision was that every deceased organ donor one day at organ donation organizations across Canada will participate in randomized clinical trials. So that was 7 years ago, and maybe we're halfway there. Today, I'm going to contrast randomized clinical trials, pragmatic trials, and platform trials at the request of Marat. And then I'll spend a few slides just focusing on platform trials, and then sum up with a couple of slides on steps to pragmatic and platform trials in deceased donor care in Canada. So a randomized controlled trial is an experiment. It randomly assigns participants to receive an experimental treatment in the experimental group or a control treatment in the control group. And the really important word here is control, because in randomized controlled trials, we as investigators take a lot of control to make sure that the experiment is assessing just the effect of that experimental treatment, and that there aren't any other biases or confounders that are going to influence the study results. And so we control what the two groups, the experimental and control group, look like at baseline. And we do that with very strict inclusion and exclusion criteria. We control the delivery of the two contrasting treatments, the experiment, very defined dose, defined period of infusion, defined time of day, or relative to the time of organ recovery, for instance. And then we define or control the control group, which is very often a placebo control, sometimes something else, but it's very specific. A lot of time and effort goes into to that as well. And then we control the similarity of concurrent care that, for instance, deceased donors in a donor intervention trial would receive, so that we want the care that they're receiving between the two groups to be identical aside from this experimental treatment. And so we protocolize a lot of other patient care, or in this case, deceased donor care in the ICU, to make sure that everything is the same between the two groups. And then finally, there's a lot of control over our outcome measurements. We typically in randomized trials will have very objective, clearly defined, explicit, and reproducible measurements of study outcomes. So that's a randomized controlled trial. And I have a case study here to share with you, which is Frederick D'Aragon's Synergy pilot trial currently underway. And here, the rationale was that there was emerging data supporting that we pre-treat neurologically deceased organ donors with calcineurin inhibitors to prevent the threats of ischemia reperfusion injury in our transplant recipients. So he went in with a host of collaborators, designed this randomized controlled trial, initially targeting 90 neurologically deceased organ donors. But the other important participants, of course, in this research is the roughly 300 organ recipients associated with those donors. And since this was a pilot trial before embarking on a broader trial at a national level, we undertook this, or plan to undertake it, in 7 intensive care units in Ontario and Quebec. But because the organs from those 7 intensive care units might go to any of the adult transplant programs in those 2 provinces, we needed to bring on side all 27 adult transplant programs in Ontario and Quebec. The experimental intervention is an intravenous dose of tacrolimus 4 to 8 hours before the scheduled organ retrieval surgery. The control intervention is an identical appearing intravenous placebo. And the clinical outcomes, particularly for a follow-up definitive study, would be delays in renal graft function in the first week post-transplantation, and delays in all graft functions in the first week post-transplantation, and then a measure of transplant graft loss after 1 year. And in the spirit of randomized controlled trials, all of these outcomes are explicitly defined and recorded by trained individuals. So that's a case study that I'll just refer to as an example as I go through this presentation. And the other thing that's important is that you understand the work that goes into putting together such a randomized controlled trial, because it's that intense work and use of resources that really commands us to look for alternative methods like pragmatic trials and like platform trials. So it all starts at the Methods Center. Might take us a few weeks or a couple of years to develop the perfect research question. And then there's the whole process of applying for funding. Sometimes we don't get funded on the first application. And then there's all sorts of authorizations from Health Canada and other regulatory bodies. Then there's training all of the individuals at donation sites, transplant sites, organ donation organizations, so many places to assist in the implementation of this work. We launch the trial. We implement the trial. It's underway right now. Eventually, we'll be analyzing and interpreting the data. And then when it's done in a randomized control trial, we shut down all that infrastructure until the next randomized trial question comes along, and then we go through that whole process again. Now, the next bullets I won't go through in such detail, but those folks at the Methods Center are teaching Methods Center research staff in all the tasks that they need to undertake with respect to research ethics board approval, study contacts, training everybody else, data transmission. They train the ICU research staff to do their very many roles in this research. They train the research staff at the transplant sites because they too have a lot of roles in this research. I didn't note here that we've also had to train up intensively a number of people at the Trail and Gift of Life Network and Transplant Quebec. And at the transplant sites, in that second final bullet, there's the issue of consent. So sometimes we actually have to train the clinicians to get involved in the consent process, as we have in the Synergy study. And in various trials, this might happen in the transplant clinic at the time of organ allocation or in the post-transplant period, or all of the above. So we do all of that for each trial, then we shut it all down, and then we repeat with the next trial that comes along. So it's very resource.
Maureen, I don't know if you can hear me, but we seem to have a problem with the connection. So if you could just hold on for 1 sec, maybe it'll come back. Oh, no, yeah. So Maureen, again, if you can hear me, we've lost connection. So just hang on a second. Okay, she may be logged out to try coming back in. Yep. Hi, Maureen, you're back? Oh, you're on mute, but you are back.
Okay, do you have my slides now?
So we can show you where we lost you if you like. Your slides are back. You're good. Yep. Thank you.
Okay, was I just finishing up here on control in pragmatic trials?
Maybe more towards the beginning of the slide.
Oh, I'm sorry. I don't know what happened. Okay.
Oh, thank you.
Okay, so pragmatic trial, clinical trial, it focuses on the correlation between treatments and outcomes in real world practice in a health system, rather than controlling all those aspects of the participant experience that I was presenting to you earlier. So that because all of that control risks rendering the trial results almost irrelevant to a real world practice. So in a pragmatic trial, less control. We do want to achieve similarity of the groups at baseline. One way to do this would be to say, okay, all NDD donors coming into this ICU are going to be eligible for this trial and will be randomized. And so there's, less finicky attention to the inclusion, like detailed inclusion and exclusion criteria, such that all donors are eligible, and that makes it much more pragmatic. There's less control over concurrent care. So in a pragmatic trial, we might not protocolize all the care that donors are getting in the ICU, but we would just have people recording that information. And already organ donation organizations are recording a lot of that information, so we could obtain it from them and describe it in a study report or manuscript. Also, in a pragmatic trial, we might achieve similarity of outcome measurements without the same degree of sort of micromanaging control. So instead of training researchers to go in and record all this data, we just get the outcomes from administrative databases like CHI in the Synergy study, the Health Canada InfoWay, hopefully in the future. And so, for instance, programs in Quebec and Alberta might define renal graft function or delayed renal graft function differently, but it's all going to come out in the wash because there's so many donors from across the country, and then we can rely on administrative databases to provide the outcomes. And then also less control in a pragmatic trial on the contrasting treatment. So for instance, there might not be a placebo control, but rather the control group would just be usual care without the experimental intervention or experimental treatment. So these are some ways that we can achieve a pragmatic trial that's really fits with the real world experience. So we can see how a drug can work in the real world, and it's more pragmatic with respect to implementation. Okay, so a platform trial is altogether different because it's more of a platform for conducting a series of experiments with the same infrastructure. So a platform trial is a prospective, disease-focused, adaptive, randomized clinical trial that compares or can compare multiple, even simultaneous interventions against a single control group. I got that from Wikipedia. And so let's just break that down. So it's disease-focused. So instead of like the Synergy study is really focused on tacrolimus, and when we answer that question, we stop the trial. A platform trial would focus on the management of deceased organ donors and have a number of experiments going on simultaneously and sequentially, and or a mix of both. And so that's a fundamental difference with a platform trial versus just a classic randomized clinical trial. This term adaptive is quite important because not all platform trials are adaptive, but you can build in statistical methods that are working behind the scenes based on all sorts of assumptions that we have to program into these statistics. But they're looking at, for instance, sample size. We might have a sample size that we're estimating for a certain experiment. Maybe it's Synergy. But if we're finding behind the scenes, the people who are privy to this information, that there's a big separation in transplant outcomes between the Synergy group and the control group, then we might find that we can do the study with a smaller sample size, or that we need a larger sample size so we can adapt the trial in real time to achieve that goal. In some of the COVID studies, there was the opportunity to, in one platform, if there were so many COVID interventions or experiments going on, if there's one that really seems to show promise, then if individuals being considered for the platform trial consent, then they can be preferentially participating in the experiment of this potentially positive or helpful intervention, and even allocated rather than one-to-one control versus the experimental therapy, but far more, a higher proportion to receive the experimental therapy so that more people are potentially benefiting along the way. So these are some of the ways that we can adapt the function of a platform trial as it's underway. And just of note, platform trials may or the experiments within a platform trial may or may not be pragmatic or highly controlled in design. Okay, so I've sort of summarized what I think are the fundamental differences between a randomized control trial, a pragmatic trial, and a platform trial, but there's other benefits to a platform trial. So one is speed. So we really, these platform trials really took off in the COVID pandemic. And without platform trials, I'd say there's no way that internationally we would have been able to address so many questions of therapeutic effectiveness. There's so many experiments undertaken in the context of national platform trials that results were coming in quickly. They went to the WHO. They were able to feed back to us clinical practice guidelines for international care that really aided us in this pandemic crisis. Efficiency is another benefit because as one experiment wraps up, the whole infrastructure doesn't collapse. We keep it going for other experiments that are underway on in the platform trial or other interventions and experiments that are about to come. So efficiency in that way is very important. I explained about how they can be adaptive. Potentially, I haven't seen this work really well as of yet, but I think it has for some in some other countries where a platform trial would have a single consent form that covers all the different experiments in the platform and a single data sharing agreement or contract between participating sites and the sponsoring site so that we don't have to have, 10 different contracts and 10 different data case report forms for 10 different experiments underway. And that really economizes on resources in so many ways. Centralized data management, we might talk about more at the workshop tomorrow, but there's a lot of ways that data management can be much more efficient in the context of a platform trial. But a huge benefit is the potential for international collaboration. And so as we embrace platform trials more and more, and research groups around the world are doing so, then, for instance, if Dan Harvey's collaborators in the UK have their own donor intervention platform trial, and we have our own, understanding that other platform trials in other countries are underway, we can unify our case report forms so that they're not just unified within a platform, but across platforms. And it makes data sharing much more efficient. And this worked extremely well, again, in the context of the COVID pandemic and different research groups undertaking very similar research. They could, their data could be very quickly harmonized and analyzed together for clinical practice guidelines. So those are a lot of the benefits and advantages, but there's really high-level needs to implement a platform trial. First of all, it takes a lot of preparation. It takes time and it takes resources. You couldn't just, think, okay, well, we've got 2 studies we want to undertake in deceased donor interventions. Let's do it in a platform trial. It would take at least a year, if not much more, of preparing a suitable platform for that. So we could get started on so many things right now. But that takes resources. And ultimately, a platform trial is going to require a sustainable funding plan because we can't build up and allow to shut down platform infrastructures the way we've been doing for just individual randomized clinical trials for years. Another critical need of platform trials is advanced expertise, and specifically advanced statistical expertise. So I would say there are at most 5 individuals, maybe 5 groups, probably 5 individuals in all of Canada who have the experience to really lend support to platform trials, and especially adaptive platform trials, because there's data simulations that are required before we launch the platform. There's all this programming to allow for adaptations and the harmonization of data at the end. We just don't have that expertise in Canada, and people are developing it in Toronto, in Vancouver, probably in other places too, but we're really behind. So most of the platform analyses are sent down to what's called the Berry Consultants, which is in a US enterprise, and they do a lot of the platform trial analyses for us. Above all else, national platform trial is going to require very strong support from a research community and a health system. So in this case, the Canadian national and provincial organ donation organizations, critical care, and transplant research communities, as Dan so nicely highlighted, and other organizations like Canadian Blood Services and the Health Canada Infoway. And as Dan also mentioned, that requires governance and really thoughtful governance. Okay, so I'm going to skip this slide. I think I've really covered a lot of that material, and we'll be getting into it in the workshop tomorrow. And just my recommendations really to steps to a pragmatic trial in Canada. And really, pragmatic trials are not all or none. You can implement some aspects to make your trial pragmatic. And so there's a menu to choose from here. We could move to trials for really generic interventions like statins in the SIGNET study, which might be applicable across organ types, and just consider all donors eligible for this trial with a few exclusion criteria only, so that will really facilitate enrollment, as they're doing in the SIGNET study, where the donor consent or the donor family consent is solicited by the specialized nurses in organ donation in the UK. We call these the organ and tissue donation coordinators in Ontario anyway. This is something we do not do in Canada for the most part, but I think we should move towards that, because even our patient partners are saying these are the people that they are, you know, most happy to talk to about anything related to organ donation after they provide their consent. Another step would be to really organize, as we have in Synergy, to obtain a lot of data from the organ donation organizations, but we could do that even more so. So instead of research coordinators in there collecting data in the ICU about how we're caring for Synergy donors, we could collect that information from the organ donation organizations if they're collecting it well. And I think year after year, the organ donation organizations across Canada are improving their job at data collection and recording and accessibility. So soon that will be much more feasible than it is now, but they're already helping us enormously in this regard. And then lastly, it will be hugely pragmatic if we collect recipient outcome data from CIHI and from the Health Canada Infoway. So even in Synergy, we're able to collect the 1 year outcome data from CIHI, but not so much for the province of Quebec. And the early post-transplant graft function in the first week, even CIHI is not going to be able to provide complete data in that regard for us. So we do have trained researchers on the ground collecting that information. And finally, steps to a platform trial. I think this will require national oversight, and I can think of so many ways that could be helpful with respect to research ethics board reviews at a national level, data safety monitoring boards for the different experiments. That would be great. But this second bullet to me is the most important step that we have to address for national platform trials, even national randomized clinical trials in deceased donor care in Canada. And it's something that nobody really anticipated and we haven't heard about yet today. But in the Synergy study, when we applied for funding, we received so much enthusiasm and support from all the organ donation organizations. Every single transplant program in the 2 provinces signed on to the grant and provided letters of support. And the vast majority, like 98% of them, allowed us to, we were able to present rounds to the transplant program, train up the research staff and some of the clinical staff on their teams for their roles in this research. And it was an enormous undertaking. And then just a year and a bit into that process, the same week that we had all of our contracts in to launch in Ontario, we found out that one of the transplant programs, which is it's a big transplant program, had some other research commitments that for which they were concerned about co-enrollment, like if the donor's in a study, but the ex vivo organs are in a different study, then how is that going to affect, like, is that going to confound any of the studies? Is that allowed? Is that conventional? And I think that the critical care world, research world, is very familiar with co-intervention in clinical trials, but not so much the transplant world. And we've learned that by connecting with a lot of transplant researchers. And unfortunately, in this case, justifiably, I'd say, because of their concerns, we had to withdraw that program from the study. As a result, that rendered infeasible, and it's too difficult to go into the details of that right now, but it rendered infeasible the work of the Trillium coordinators who are allocating the organs and involved in our research consent process, so that ultimately we had to shut down the trial after all that year of work in Ontario. So it is still underway in Quebec. We see this as a setback and a blip in the road, but it's something that we absolutely have to contend with in advancing to national deceased donation research. And then my last bullet here is I would recommend that for a platform trial, we start narrowly and then build on it. So rather than putting all deceased donors into a platform trial, or even all DCD donors or NDD donors, that we start with a single common organ type, like maybe start with a platform for experiments in NDD kidney donors, get a bit of experience using a platform, and then grow from there to include DCD and other organ types and all donors across Canada. So sorry for the technical difficulties, but that's my last slide, and I'll sign off so that we can move to questions.
Thank you, Maureen. And I'm not sure why the switch, but I'm asking to moderate. We don't have a lot of time, so maybe just time for one question, if anybody has one question. And I'll just, we'll wait for the speaker to come up to the mic. I'll borrow words of Jennifer that, you know, there's all the science surrounding trials and, you know, regular trials, pragmatic trials, platform trials. And we can create all the strategy in the world, but then we're going to hit the floor and we're going to be met with culture, and culture is going to eat us for breakfast. So unless we address it nationally, I think, and try to change the culture to enable trials to proceed through transparent governance and all the things mentioned, I don't think we can move forward. So we will work on that. We can start working on that tomorrow at 7 a.m. So one question.
So I just wanted to thank all the speakers, and I'll be there at 7 a.m. to participate in that session as well. I'm really looking forward to it. I did want to just maybe sort of add to what Maureen was saying. It's this concept of platform trials is really, really interesting. But I think I would invite, since we're the CDTRP, to invite the donation and transplantation platform type of trial. So this isn't just about getting consent for the donors on the donor side of it, but also from the recipients on the recipient side of it, so that we can actually study the continuum of donation through into transplantation. It's not going to be lost on anyone that what happens on the donor side may affect what we do on the recipient side in terms of management, and especially when we start talking about, tweaking the immune system in the donor kidney versus the recipient at the time of transplantation. And I think it's going to be really important. And the work that Maureen and your group has done to engage with the transplant centers, I think, is pivotal to that process, because I think it's going to be together that we're actually able to make something big happen here. Thank you.
Thank you for that comment. Maureen, you want to respond, and then we'll wrap up?
Well, just to say, like, we have been just delighted with the support from the transplant community, including those that weren't able to ultimately participate in Synergy. And I know that we can work through this, and I'm really looking forward to the workshop tomorrow. Thanks, Marat.
All right. Thank you very much, everybody.